Fine, let's start. Number 1. Dolly the sauropod and the air sac infection. Picture a long-necked sauropod in what is now Montana. The time is the late Jurassic, about 150 million years ago. The animal stretched roughly 18 meters from nose to tail. That is the footprint of a detached house. It walked the floodplains of the Morrison Formation, browsing on conifers and ferns. And it was sick. Deep inside its neck, something was eating into the bone. We know this animal by a museum number. The specimen is cataloged as MOR 7029, held at the Museum of the Rockies. The team gave it a friendlier name. They called it Dolly, after Dolly Parton. The case was published in the journal Scientific Reports in February of 2022. The lead author was the paleontologist Cary Woodruff. He worked with the respiratory disease researcher Ewan Wolfe, the sauropod specialist Matthew Whittle, and the veterinary radiologist Sophie Dennison. 3 of Dolly's neck bones carried strange growths. These were irregular lumps of bone that had no business being there. They were not healed fractures. They were not tumors. And they sat in a very specific place. They formed exactly where soft air sacs would have pushed into the bone. Here is the key to the whole story. Sauropods breathed like birds. They had a system of air sacs connected to their lungs. Those air sacs invaded the skeleton and filled the bones with pockets of air. Scientists call this skeletal pneumaticity. It made these giants lighter. It also gave disease a way in. When the researchers ran the bones through a CT scanner, they saw the texture of the lesions. The growths looked like unripe cauliflower. That pattern matched a known process. An infection had spread out of the lungs, into the air sacs, and then into the bone itself. The medical term is air saculitis. It is an infection of the air sacs. The closest living comparison is a disease called aspergillosis. It is caused by a fungus named Aspergillus. It infects the air sacs of modern birds and reptiles. Birds with this disease lose weight. They run fevers. They cough and breathe with effort. Dolly may have suffered all of these things. We almost never see disease like this in fossils. Soft tissue rots away. The only reason we can study Dolly's illness is that it reached the bone and left a permanent mark. Everything else is long gone. So, what exactly was the pathogen? Here the science splits. The mainstream view holds that this was a fungal infection in the style of aspergillosis, breathed in from rotting plants. A second view suggests a bacterial infection, perhaps following another illness or injury. A third, more cautious idea admits the truth. We cannot name the exact organism from bone alone. It could have been fungal, bacterial, or something else entirely. Whatever it was, it deformed the spine of one of the largest animals of its age. A microbe hollowed the neck of a creature the size of a house. And if a microbe could do that to the living, imagine what waited for the dead. Number 2. The bone-eating worms of the Cretaceous seas. Now sink to the floor of a Cretaceous ocean. A plesiosaur has died. Its body drifts down through the dark water. This was a marine reptile over 10 meters long, with a barrel chest and paddle limbs. When it lands on the seabed, it does not simply vanish. It becomes a feast, and the strangest guests at that feast eat the bones. These animals are worms of the genus Osedax. The name means bone devourer in Latin. They were first discovered in the year 2002. A research team from the Monterey Bay Aquarium Research Institute found them on a dead gray whale. The carcass lay about 2,900 meters down in Monterey Canyon. The worms were described in the year 2004 by the scientists Greg Rouse, Shauna Gafridi, and Robert Vreinhoek. The biology of Osedax is almost unbelievable. The adult females have no mouth. They have no gut. They have no anus. Instead, they grow root like structures that bore down into bone. Inside those roots live colonies of symbiotic bacteria. The bacteria break down the collagen and the fats locked inside the bone. The worm secretes acid to dissolve the mineral. Together, they hollow the skeleton from within. There is more. The males are tiny. They are microscopic dwarfs that live inside the tubes of the females. A single female can carry dozens of them. It is one of the most extreme size differences between sexes in the animal kingdom. The worms leave a signature. As they bore, they carve a distinctive cavity. There is a narrow opening at the surface that widens into a bulbous chamber below, often branching. These cavities survive as trace fossils, even when the soft worm itself does not. In the year 2015, the scientists Sylvia Denise and Nicholas Higgs published a discovery in the journal Biology Letters. They found Osedax-style borings in fossil bone from the United Kingdom. The bones belonged to sea turtles and plesiosaurs. They were about 100 million years old. This rewrote the timeline. Whales did not exist 100 million years ago, so the worms could not have evolved to eat whales. They must have first fed on the carcasses of giant marine reptiles. Only later, after the dinosaurs died out, did they switch to whales? This raises a haunting possibility. Researchers call it the Osedax effect. If these worms destroyed marine reptile skeletons for tens of millions of years, they may have erased countless fossils. The very record we use to study the past has gaps the worms ate. How certain is all this? The mainstream reading says Osedax evolved on reptile bone falls and later moved to whales. A second, more cautious view notes that some Cretaceous borings might come from other organisms, not Osedax. A third idea goes further. It suggests the deep sea bone fall ecosystem ran continuously through the entire age of dinosaurs. These worms consume the dead, not the living, but they truly hollow the bones of giants. And while they worked from the outside, other parasites attacked from within the blood. Number 3. The blood parasites of the dinosaurs. Imagine a small biting insect landing on the skin of a dinosaur. It pierces the hide. It fills with blood. Then a drop of tree resin falls and traps it forever. 99 million years later, we crack open that amber and look inside. And in some cases, we find more than the insect. We find what was rotting in its gut. Most of this work comes from one researcher above all, George Poinar Jr. of Oregon State University, often working with Roberta Poinar. They are specialists in organisms preserved in amber. In the year 2004, Poinar described a parasite called Paleoleishmania proteris. He found it inside a fossil sandfly preserved in Burmese amber, about 100 million years old. The parasite is a relative of modern leishmania. That is the organism that causes the disease leishmaniasis in people today. Poinar reported that he could see different life stages of the parasite inside the insect. The next year, in 2005, Poinar worked with the parasitologist Sam Telford. Together, they described Paleohemoproteus burmachis. They found it inside a biting midge. This parasite belongs to a group called the Hemosporidians. That is the same broad family that includes Plasmodium, the cause of malaria. So the picture is striking. The insects that carried these parasites are blood feeders. Their living relatives bite reptiles and birds. If their Cretaceous cousins did the same, then dinosaurs could have caught malaria-like diseases and leishmania-like diseases. They could have run fevers. They could have suffered anemia. The amber comes from a single rich source. It is the Hukong Valley of northern Myanmar. It dates to about 99 million years ago, in the middle of the Cretaceous. The preservation is so fine that single-celled parasites can be seen inside the insects. Poinar built this into a larger argument. In the year 2008, he and Roberta Poinar published a book called What Bugged the Dinosaurs. In it, they proposed that insect-borne diseases were a slow drain on the dinosaurs, one factor among many in their long decline. Now, for the honest part. No parasite has ever been found inside an actual dinosaur. The whole case is built by inference. We have the vector, the insect. We have the parasite inside it. We know the feeding behavior. We connect the dots. This is where the theories split. The mainstream view accepts that blood parasites existed and likely infected dinosaurs through insect bites. A second, skeptical view warns that the tiny structures inside amber insects might be gut contents or preservation artifacts, not parasites at all. A third view is Poinar's own. He argues disease helped push the dinosaurs toward extinction. Most paleontologists disagree and rank disease far below the asteroid impact and the great volcanic eruptions. These parasites are some of the only glimpses we have of microscopic disease from the dinosaur world. And the insects that carried them were preserved too. One of them was caught in the act, still clinging to its host. Number 4. Dracula's terrible tick. Some fossils are arguments. This one is almost a photograph. It is the single most direct piece of evidence we have for a parasite feeding on a dinosaur, and it survived inside a drop of amber. The discovery was published in the year 2017 in the journal Nature Communications. The lead author was the paleontologist Enrique Penalver. The fossils came once again from Burmese amber, about 99 million years old. The first specimen is a hard tick. Its species name is Corna palpatum burmanicum, and it is gripping something. It is holding onto a single dinosaur feather. This is the first time a tick has ever been found physically attached to evidence of its dinosaur host. The feather is the clue to the host. It is a modern style feather. But true birds, in the sense we mean today, are not part of this picture at that time and place. So the feather is read as belonging to a feathered dinosaur, a small meat-eating theropod. The team also found a brand new kind of tick. They named a new genus and species, Dinocraton draculae. The name means Dracula's terrible tick. They chose it because one specimen was swollen with blood. That engorged tick had ballooned to about 8 times its normal volume. It had recently fed. Now hold your excitement, because here is the limit. The blood inside could not be analyzed. No host DNA could be recovered. So the idea that this is dinosaur blood is an inference from the surroundings, not a tested sample. There were more clues. 2 of the Dinocraton ticks were found locked together. The researchers think they shared a single host, probably a nest, and the ticks carried tiny hairs on their bodies. Those hairs match the larvae of skin beetles. Those same beetles are known to live in the feather-lined nests of feathered dinosaurs. The web of evidence tightens around a nesting dinosaur. Ticks are ancient. They are among the oldest blood-feeding parasites, with a record reaching into the Cretaceous and likely far beyond. The reason we can see this scene at all is luck. Resin froze the tick, the feather, and the beetle hairs together in one moment. There is a modern echo. Ticks today carry Lyme disease and many other illnesses. If ticks are this old, then tick-borne disease may be just as old. How do experts read this fossil? The mainstream view holds that the tick parasitized a nesting feathered dinosaur. A more cautious view accepts the feather and tick link, but stresses that the blood source is unconfirmed. A third, speculative view imagines dinosaurs suffering tick-borne diseases we may never be able to detect. There is also a hard truth around this fossil. Burmese amber is mined in the Kachin region of Myanmar, an area torn by conflict. The science carries an ethical weight. A tick a few millimeters long, feeding on an animal that could dwarf a house. And in the same amber, even smaller stowaways were chewing the feathers themselves. Number 5. The feather-chewing insects. Zoom in closer still. Past the tick, down to the surface of the feather itself. There, almost too small to see, something is eating. These are insects barely a fraction of a millimeter long. and they mark the dawn of a war that still rages on every bird alive today. The discovery was published in the year 2019 in the journal Nature Communications. The lead author was the researcher Taiping Gao. The species was named Mesophthyrus engeli. Once again, it came from Burmese amber, about 99 million years old. The specimens are tiny insect nymphs. They are under 1 millimeter in length. They have no wings. They have stout legs built for gripping, and they have chewing mouth parts. Everything about their bodies points to a life spent clinging to a host and eating it. The feathers in the amber tell the rest. They show ragged, irregular damage. That damage looks exactly like the work of modern feather lice on living birds. The insects were chewing the feathers. The researchers presented Mesophthyrus as the earliest known example of insects feeding on feathers. It pushed that behavior back by tens of millions of years. And there was not just one insect. Several nymphs were preserved together on the feathers. This was a small infestation, not a lone wanderer. The host cannot be named precisely. It was a feathered dinosaur, or possibly a very early bird. What matters is the pattern. As feathers evolved across the dinosaur family tree, parasites evolved right alongside them to exploit those feathers. The modern comparison is direct. Chewing lice in the order Pithoraptora live in the plumage of birds today. They damage feathers. They irritate the skin. They can open the door to other infections. Mesophthyrus appears to have done the same to dinosaurs. Our picture is incomplete. Only the young nymphs were found. The adult form is unknown. The full life cycle is a mystery. And some experts debate whether Mesophthyrus is truly a louse or a different early kind of insect. This is where the readings divide. The mainstream interpretation treats it as a genuine feather-feeding parasite, co-evolving with feathered dinosaurs. A second view suggests it might be a non-louse insect that simply ended up near the feathers. A third, behavioral idea proposes that feathered dinosaurs preened and groomed themselves to fight these pests, just as birds do now. Tiny insects, exploiting animals that in some cases were enormous. Together with Dracula's tick, this find makes Burmese amber the richest window we have into the parasites that lived on dinosaurs. Now leave the dinosaurs behind. The same parasitic strategies survived the ages. Tens of millions of years later, they colonized the giants of the Ice Age. Number 6. The botfly inside the mammoth. Travel forward in time to frozen Siberia. A woolly mammoth lies preserved in the permafrost, where it has rested for thousands of years. Its flesh is intact. Its stomach still holds its last meals. And inside that gut, scientists found something alive in form, if not in fact. They found the larvae of a parasite that lived inside the mammoth. The mammoth itself was a giant. The woolly mammoth, Mammuthus primigenius, stood over 3 meters at the shoulder. It could weigh 6 tons or more. This was Ice Age megafauna at house scale. The parasite is a botfly of the genus Coboldia. The species recovered from mammoth remains is Coboldia rusinovi. It is associated with the Soviet entomologist named Grunin, who worked on these flies in the 1970s. The larvae develop inside the digestive tract of large mammals. We have a perfect living comparison. The genus Coboldia still exists. One species, Coboldia elephantis, lives in the stomachs of modern elephants, and the most familiar relative of all is Gasterophilus, the horse stomach botfly. Anyone who has kept horses knows it. The life cycle is grim and elegant. The adult fly lays eggs or larvae on the host's body. The host swallows them. The larvae then anchor to the wall of the stomach. There they feed and grow before passing out of the body to pupate. Inside the mammoth, they did exactly this. One famous specimen sets the scene. The Berezovka mammoth was recovered in the year 1901 from the banks of the Berezovka River in Siberia. Its stomach still held undigested vegetation. Frozen mammoths like this preserve not only the animal, but the soft-bodied parasites within it. This is the gift of permafrost. The same deep freeze that saves mammoth flesh also saves its parasites. From a single frozen gut, scientists can reconstruct an entire community of organisms. The effects on the host would have been real. In modern elephants and horses, heavy botfly infestation causes ulcers, irritation, and blockages in the gut. Mammoths likely suffered the same. How do we interpret these larvae? The mainstream view treats Coboldia as a normal stomach parasite of mammoths, exactly as in living elephants. A second cautious view notes that some larvae might be post-mortem colonizers rather than lifelong residents. A third speculative idea suggests that parasite loads added stress to mammoth populations already in decline near the end of the Ice Age. There is a modern twist. As the permafrost thaws, more frozen mammoths are emerging from the ground, and their parasites with them. These are Cenozoic parasites, far younger than the dinosaur examples. They carry the story from deep time into the Ice Age. From the soft belly of a mammoth, return now to the airways. The strangest parasite of all does not live in the gut. It lives in the lungs. Number 7. The tongue worms of the giant crocodiles. Some parasites are so strange they barely seem real. These are the tongue worms. They live as adults inside the lungs and airways of living animals. And to find their giant hosts, we have to imagine crocodiles the size of buses. The proper name for the group is Pentastomida. There are about 140 living species. Most of them live in the breathing passages of vertebrates, and they have a special fondness for reptiles. Here's the first surprise. They look like worms, but they are not worms. Molecular and developmental evidence shows they are highly modified crustaceans. They are distant relatives of the fish lice, a creature in the same broad group as crabs and shrimp, living inside a lung. Their bodies are ringed, like a series of stacked segments. The head carries 2 pairs of hooks. Those hooks gave the group its name. Pentastomida means 5 mouths, because the hooks and the mouth together look like 5 openings. The hooks anchor the parasite into the host's tissue. The living relatives infect reptiles directly. Genera like Armilifer and Linguatula are well known, and the cebacids, such as Sebeckia, live in the lungs and airways of crocodiles. The fossil record of this group is astonishingly deep. Tiny larval forms have been recovered from rocks in Sweden that are over 400 million years old. Then there is a true landmark. In the year 2015, the scientist David Siviter and his colleagues described a fossil named Invavita piratica. It came from the Herefordshire deposit in England, about 425 million years old, and it was preserved attached to its host, a small shelled animal called an ostracod. It is one of the oldest parasites ever found, still clinging to the thing it fed on. Younger fossils of sebecid tongue worms are found alongside crocodile remains. This ties the group firmly to the crocodile line through deep time. So it is very likely that giant prehistoric crocodiles carried these parasites in their lungs. And what giants they were. Consider Sarcosuchus imperator, the so-called supercroc. It was described in detail by the paleontologist Paul Sereno in the year 2001. It reached around 11 to 12 meters long and lived in the Cretaceous of what is now Niger. Consider also Dinosuchus, a late Cretaceous giant of North America, around 10 meters long, big enough to hunt dinosaurs. We have no fossil of a tongue worm inside one of these crocs. The adults live in soft lung tissue, which almost never fossilizes. The case rests on the group's great age and its tight bond with reptile hosts. So the theories line up this way. The mainstream view says giant crocodiles almost certainly carried lung-dwelling tongue worms. A cautious view stresses that without a body fossil inside a croc, the burden is inferred, not proven. A speculative view wonders whether heavy infestations stressed these semi-aquatic giants. A parasite a few centimeters long lodged in the lungs of a 12-meter crocodile. And if tongue worms reach back 400 million years, tapeworms reach back even further. Number 8. The oldest tapeworm eggs. Now, go back deeper than any dinosaur, deeper than the giant crocodiles, to a time before almost everything in this video. The evidence is humble. It is a piece of fossil dung, but what it holds rewrote the history of parasitism. The discovery was published in the year 2013 in the journal PLOS1. The lead author was the paleontologist Paula Dencian-Diaz. The title said it plainly: Tapeworm eggs in a 270 million year old shark coprolite. A coprolite is fossilized dung. This one came from the Rio do Rastro Formation in the Parana Basin of southern Brazil. It dates to the Permian period, the chapter of Earth history just before the dinosaurs. The shape of the dung is a clue to who made it. It is spiral. That spiral form points to a shark or a similar fish with a spiral valve in its intestine. When researchers sliced the coprolite thin and put it under the microscope, they found a surprise. There was a tight cluster of about 93 small oval eggs. The team identified them as the eggs of a tapeworm. At least one egg appears to hold a developing larva inside. Some show the structure of a shell. The significance is enormous. This single find pushed the record of tapeworm parasitism back by roughly 140 million years. Before this, the oldest solid evidence of tapeworms came from much later, in the age of dinosaurs and after. The Permian eggs blew past all of it. It tells us something profound. Complex internal parasitism, a soft-bodied worm living hidden inside a vertebrate, was already happening a 250 million years ago. This was long before any dinosaur drew breath. There is a reason the eggs are all we have. Tapeworms are soft. Their bodies essentially never fossilize. The only realistic way to glimpse their deep history is to find their eggs, sealed inside fossil dung. The life cycle leaves its mark. The host shark almost certainly picked up the tapeworm by eating a smaller animal that carried it. That's exactly how modern tapeworms move between hosts. The timing matters too. The Permian period ended about 252 million years ago in the largest mass extinction in the history of life. These parasites lived in the world right before that catastrophe. How is the find read by scientists? The mainstream view accepts these as genuine Permian tapeworm eggs, proof that the lifestyle is at least 270 million years old. A cautious view, as with all microfossils, keeps the door open to other interpretations, while agreeing the tapeworm reading is the strongest. A more speculative idea wonders whether such parasites rode through the Great Extinction alongside their hosts. This is the deepest anchor of our whole story, and from these ancient seas, we now leap forward to the most famous predator that ever lived, and the parasite that may have scarred its jaw. Number 9. The parasite that scarred the tyrant king. Everyone knows this animal. Tyrannosaurus rex, the tyrant king. It reached around 12 to 13 meters long and weighed up to 9 tons. It was a house on legs with teeth like railway spikes, and it may have been brought low by something a single cell across. The most famous individual is named Sue. The specimen number is FMNHPR 2081. Sue is the largest and most complete Tyrannosaurus rex ever found, on display at the Field Museum in Chicago, and Sue's lower jaw has holes in it. These are smooth-edged, roughly circular holes punched into the bone. For years, people had a dramatic explanation. They said these were battle wounds, bite marks from fights with other tyrannosaurs, or they blamed a simple bacterial infection of the bone. Then, in the year 2009, came a new idea. The researchers Ewan Wolf, Stephen Salisbury, John Horner, and David Verricchio published a study in the journal PLOS1. Its title was direct. A common avian infection plagued the tyrant dinosaurs. Their argument was this: the smooth-edged holes in Sue's jaw look like the damage caused by a parasite called Trichomonas gallini. It is a single-celled organism, a protozoan. Today, it infects birds. Pigeons and doves carry it. The hawks and falcons that eat those birds catch it too. Falconers call the disease frounce. In pigeons, it is called canker. The mechanism fits the bones. The infection creates cheesy lesions in the mouth and throat. Those lesions can erode down into the jawbone. The result is a smooth margined hole, exactly like the ones in Sue. And it was not just Sue. The team examined many tyrannosaurs. They found similar lesions in roughly 1 in 10 of them. This was a widespread problem, not a single unlucky animal. The consequences could be deadly. A severe infection swells the throat. It makes swallowing and breathing difficult. The researchers suggested that heavily infected tyrannosaurs might have slowly starved to death, unable to eat. The biology makes sense. Tyrannosaurs sit on the branch of the family tree that leads to birds. Birds are the modern hosts of Trichomonas. So a bird-style infection in a tyrannosaur is entirely plausible. The parasite may have spread through cannibalism, through face biting in fights and courtship, or through shared feeding on carcasses. We know tyrannosaurs bit each other in the face because the healed wounds are there in the bone. Now, the theories. The mainstream view holds that an avian-style protozoan infection caused the lesions. A second view argues that other microbes or other diseases could produce similar holes. A third view is the older one, which still says the holes are healed bite wounds from rival tyrannosaurs. The king of predators, perhaps undone by the same kind of parasite that infects a city pigeon. From the apex hunter, we turn at last to the gentle giants of the Ice Age. Number 10. The worms inside the giant ground sloths. End the journey in a dry cave in the American Southwest. Pleistocene sunlight slants through the entrance. On the floor lies a thick layer of ancient dung. It was left by ground sloths, huge shaggy herbivores that vanished from the earth around the time the first humans walked these lands. And in that dung are the eggs of the worms that lived inside them. The ground sloths were giants. The Shasta ground sloth, Nothrotheriops shastensis, was roughly the size of a large ox. Its South American cousin, Megatherium, was far larger still, over 4 meters long, on the scale of an elephant. These were house-sized animals. The dry caves of the Southwest preserve their droppings beautifully. 2 sites stand out. There is Rampart Cave in the Grand Canyon and Gypsum Cave in Nevada. The dung found there ranges from about 11,000 to over 40,000 years old. The preservation is remarkable. The coprolites still hold plant fragments, pollen grains, and parasite eggs. Under the microscope, researchers found the eggs of intestinal worms, including roundworms, that lived inside the sloths. The eggs are identified by their shape, their size, and the structure of their shells. This is the standard method of studying ancient parasites in dung. The same story plays out in South America. Coprolites of Darwin's ground sloth, Mylodon darwinii, have been studied from the famous Mylodon Cave in Chilean Patagonia. The plant remains set the scene. The sloths ate desert plants like Mormon tea, globe mallow, agave, and saltbush. That was the world their parasites shared. There is a note of loss in this story. In the 1970s, a fire damaged part of the precious dung deposit in Rampart Cave. A record tens of thousands of years in the making was harmed in moments. The meaning is clear. Finding these eggs confirms that Ice Age megafauna carried the same broad groups of worm parasites that infect living herbivores today. When the ground sloths went extinct, around 11 to 12,000 years ago, their parasites died with them. The dung is the only record we have left. Notice the contrast in preservation. Mammoths were saved by freezing. These sloths were saved by dryness. two different paths to the same gift. How do scientists read the eggs? The mainstream view treats them as genuine intestinal worms of the sloths, preserved as eggs in the dung. A cautious view allows that some eggs could come from material the sloth ate, rather than from its own gut. A speculative view wonders whether parasite burdens added to the stress of the Great Ice Age extinction. These sloths overlapped with early humans. People hunted them. And so the story closes where the deep past meets our own, from Permian seas to caves that human feet have walked. Step back now and ask a simple question. How can we possibly know any of this? Parasites are mostly soft and small. They rot. They should leave no trace at all. And yet we have 10 cases. The answer lies in a detective's toolkit. Amber is the first tool. Tree resin traps insects and hardens, preserving them in 3 dimensions, down to the cell. That is how we see the blood parasites, the Dracula tick, and the feather-chewing insects. Coprolites are the second tool. Fossil dung traps the eggs of gut parasites. That is how we found the Permian tapeworm and the ground sloth worms. Bone lesions are the third tool. When a disease reaches the skeleton, it can leave a permanent scar. That is how we read Dolly the sauropod and Sue the tyrannosaur. Trace fossils are the fourth tool. Borings and tunnels reveal organisms like Osidax, even when the worm itself is gone. Frozen ground and dry caves are the fifth tool. Permafrost saved the mammoth's botfly. Dry caves saved the sloth's dung. Then come the instruments. CT scanning sees inside bone without breaking it. It read Dolly's vertebrae and the shape of the worm borings. Thin sectioning and the microscope reveal eggs hidden in dung. There is also the method of comparison. Scientists match ancient damage to known modern diseases. That is exactly how Sue's jaw was diagnosed, by comparing it to a pigeon's illness. Every tool has a blind spot. That is why so many of these cases stay labeled as interpreted, not proven. Preservation bias means we see only a sliver of the parasites that once existed. The same names appear again and again in this field. George Pointer, Carrie Woodruff, Ewan Wolfe, the Civeter team, Paula Dencian-Diaz. And new imaging keeps reopening old fossils to fresh diagnoses. With the tools in hand, a pattern begins to emerge across all 10 cases. Look at the 10 stories together, and a single theme rises up. For more than 270 million years, parasites have shadowed their hosts, step for step. As feathers evolved, parasites evolved to exploit them. The feather-chewing insects and the feather-clinging tick are two halves of the same arms race. As bird-style air sacs evolved, air sac infections became possible, and Dolly paid the price. As crocodiles arose, tongueworms colonized their lungs. As elephants and their kin evolved, host-specific botflies followed them into the gut. As giant marine vertebrates filled the seas, bone-eating worms followed the bodies down, from reptiles to whales. The lesson is humbling. Size offers no protection. A bigger host is simply a bigger habitat. The largest animals that ever lived were also the largest homes for the smallest. Notice too that these parasites came from utterly different branches of life. Tongueworms are crustaceans. Tapeworms are flatworms. Ticks and lice are insects. Blood parasites are single cells. Each one invaded the parasitic lifestyle on its own. They arrived at the same strategy by separate roads. The arms race ran both ways. Hosts fought back with grooming, with preening, with immune defenses, and the niches kept reappearing. When one giant went extinct, another took its place, and the parasites moved in once more. The deep sea bonefall community is the clearest example. It ran without a break from the marine reptiles straight through to the whales of today. Some parasites changed less than their hosts did. The tongueworms and the tapeworms are ancient living fossils, their body plans almost frozen in time. If parasites are this pervasive, A darker question follows. Did they ever help kill the giants off? It is a tempting idea. Tiny enemies, working in the dark, dragging down the titans. So let us weigh it honestly. George Poinar made the boldest version of the claim. He argued that insect-borne diseases were a slow stressor in the long decline of the dinosaurs. It is a provocative thesis, but it runs against the dominant explanation. The mainstream view of the dinosaurs' end is clear. About 66 million years ago, a massive asteroid struck the Earth at Chicxulub in what is now Mexico. Alongside it, enormous volcanic eruptions in India, the Deccan Traps, stress the climate. These are the heavyweights of the extinction debate. The problem with the disease idea is testability. Disease leaves almost no fossil trace. That makes its role easy to imagine and hard to prove. It is simple to overstate. For the Ice Age giants, the leading suspects are different. They are climate change and human hunting. The debate over how much humans drove the extinctions is fierce and still unsettled. Where could parasites fit? They could plausibly worsen the stress on a population already in trouble. Trichomoniasis may well have killed individual tyrannosaurs. Dolly's infection may have helped kill one sauropod, but the death of an individual is not the extinction of a species. There are points on both sides. In favor of disease, we know that pandemics can devastate populations that have never met a pathogen before. Against it, parasites and hosts usually settle into a balance over evolutionary time. They rarely wipe each other out. So what is the fair verdict? No evidence raises parasites above the asteroid, the volcanoes, the changing climate, or human hunters. The honest reading is that parasites were, at most, a small background term in a much larger equation, a stressor, not a cause. Whatever their role back then, one thing is certain. The descendants of these parasites are very much alive today. The deep past is not as distant as it feels. Trace each of these ancient parasites forward, and you arrive in the present day. The ticks like Dinocraton have living heirs. Modern ticks carry Lyme disease and many other illnesses. The protozoan that scarred Sue's jaw, Trichomonas galeni, still causes frounce and canker in pigeons and birds of prey right now. The botflies that lived in the mammoth still infest elephants and horses. The tongue worms still coil in the lungs of living crocodiles, and they occasionally infect people too. The bone-eating Osedax worms are not a fossil curiosity. They are hollowing whale bones on the seafloor at this very moment. The blood parasites in the Hemosporidian group are still with us, and they include Plasmodium, the cause of human malaria. Tapeworms remain among the most widespread parasites of vertebrates on the planet. And feather lice still ruin the plumage of birds, exactly as they did to feathered dinosaurs. This continuity is the real point. These are not exotic relics. They are the ancestors of the diseases we manage today. Studying them helps us understand how modern diseases emerge and spread. There is a timely thread here, too. As permafrost thaws, scientists ask hard questions about what ancient organisms might wake with it. The line between host and parasite has shifted again and again across time, from reptiles to birds to mammals. There is even a conservation angle. Parasites are part of biodiversity. They are threads in the web of every ecosystem, not just villains to be erased. The deep past, it turns out, is a guide to the parasitic present, which leaves one last thing to say about the giants and their unseen passengers. There is a single thread running through all 10 stories. Size is no shield against the microscopic. The largest animals that ever lived shared their bodies with the smallest. We see only what preservation allows. A drop of amber, a piece of dung, a scarred bone, a body locked in ice. Everything else is gone. Most ancient parasites left no trace at all, and they are lost to us forever. Each fossil parasite we do have is a frozen moment, a single still frame of a relationship that played out over millions of years. The giants are extinct. The strategies that fed on them endure. The worms, the ticks, the flies, the single cells, all of them found new hosts and carried on. The open questions remain, and they are good ones. Which pathogens truly killed? Which giants? And how often? How much of the marine fossil record the bone eaters quietly erased? Whether the dinosaur blood parasites were really transmitted or merely preserved. Paleoparasitology is a young science. New imaging will reopen old fossils, and old fossils will give new answers. The deepest reframing is this. Every giant was not a single animal, but a moving ecosystem, a world unto itself, carrying countless smaller lives. Even the king of the dinosaurs could be undone by a single cell. The arms race that played out across deep time has never ended. It continues inside every living animal, right now, including the one reading these words. And so we leave them as we found them. House-sized creatures, walking and swimming through the ages with their tiny hidden passengers along for the ride.